students, welcome back to Plus One chapter Biomolecules, and our today's topic is protein. In our last class, we have discussed the structure and properties of various amino acids. So, you know that protein is a biomacromolecule and it is a polymer of amino acids. Many amino acids join to form protein. So, our today's biomolecule is protein, which is a macromolecule, and sure, it is made up of number of amino acids. So, we can say protein is a polymer of amino acids. Okay, right? Then, tell me, what is the monomer of protein? So, protein is made up of amino acids. So, monomeric units of protein are amino acids. Okay, then listen carefully here. A protein is always heteropolymer. What do you mean by heteropolymer? A protein is always heteropolymer. A homopolymer. Example for a homopolymer is cellulose, glycogen, starch. It is made up of only glucose. Okay, starch is made up of only glucose. If starch is broken down, we will get only glucose. Then starch is a homopolymer of glucose. Whereas there is no protein which is a homopolymer. All proteins in the world are heteropolymers. That means it is made up of different types of monomers. I mean different types of here monomers are amino acids. So protein is made up of different types of amino acids. So we can say what? Protein is a heteropolymer of amino acids. So what is a protein? A protein is a heteropolymer of amino acids. Is it okay? So in order to form a protein, what should be there? There should be many amino acids. Then where are the proteins formed? In any type of organism. Whether it is eukaryotic or prokaryotic, it consists of the cell organelle ribosome. Okay. So you know the fact, and the synthesis of protein takes place within ribosomes. So with the help of RNA. Okay. So protein synthesis takes place in ribosomes with the help of RNA. RNA is guiding this protein synthesis. So for the protein synthesis, what is required? It requires number of amino acids and also various types of RNAs, and takes place within the ribosomes. So what we have discussed about the protein? Protein is a heteropolymer of amino acids. Different types of amino acids join to form protein. Okay. Then protein synthesis in our body, plant body, prokaryotes body, it takes place within the cell organelles ribosomes and with the help of RNA. Okay. Then another way, proteins are always polypeptides. Okay. A protein is formed by joining many amino acids. Let's have a representation. So one amino acid. Last class you have studied the structure of amino acids. Okay. Then here a group. Then the second amino acid. Okay. Just representing second amino acid. These two amino acids and again the next amino acids are joined by a bond called peptide bond. Okay. One amino acid is joined to the next amino acid in the protein structure in order to form the protein by peptide bonds. What is a peptide bond? So here the peptide bond is formed between the carbon and nitrogen. Carbon of one amino acid and nitrogen of the next amino acid. I am just splitting the hydrogen, NH, I am writing here. Then just listen here. One molecule of water is eliminated. Okay, from here, one molecule of water is released. Okay, between this carboxylic acid group and amino group. Okay, what happens? One molecule of water is eliminated. Okay, then what is left there? I'm just modifying here. So there will be CO, carbon, oxygen, here CO, here will be NH. What is left here will be carbon and oxygen, here nitrogen and one hydrogen. And here the bond is formed. Actually, the bond is formed between carbon and nitrogen. So the representation I will do like this. So the carbon will be bonded to oxygen. Here is the carbon. Okay. The nitrogen will be bonded to hydrogen. The peptide bond is between carbon and nitrogen. Okay. So this way we represent the peptide bond. And what is this bond called? This is peptide bond. So just understand, in order to form protein, one amino acid is joined to next amino acid by means of peptide bond, CONH bond. The bond is between carbon and nitrogen of the first and second amino acids. Okay? And similarly, in order to form a protein, a long chain of protein, many amino acids should be joined. A long chain of amino acids. So it consists of number of peptide bonds. We can call protein as a polypeptide. Okay? Protein is nothing but a polypeptide. So we have discussed protein is a heteropolymer of amino acids. Different types of amino acids are joined. And protein is formed inside the ribosomes, and proteins are polypeptides consisting number of peptides. Okay, what is drawn here can be called as a dipeptide because two amino acids are joined by a peptide bond. This is called a dipeptide. Okay, then I think it's clear what is a peptide bond, how is it formed. It is between carboxylic acid, COH group of one amino acid and amino group of next amino acid by the removal of one water molecule. The bond is formed. Okay, then let us come to the details of protein structure. So anyway, you know that proteins, there is no protein in the world which is a homopolymer, and you also know that protein is an abundant biomolecule in the world. Okay, which is the most abundant protein in the world? The most abundant protein. It belongs to the plants. Okay, the most abundant protein in the world is an enzyme. It is rubisco. Okay, its name is ribulose bisphosphate carboxylase oxygenase. It's an enzyme. You are going to study this in photosynthesis chapter. Okay. So, rubisco is the most abundant protein in the world. It belongs to plants. Similarly, which is the most abundant protein in animals? The most abundant protein in animals is collagen. Okay? The most abundant protein in animals' body. This animal world, it is collagen. In plant world and whole world, it is rubisco. Okay, rubisco is an enzyme. So, in order to form proteins, our body should get amino acids. Some amino acids can be produced within our body itself, whereas some amino acids surely we should supply through our diet. The amino acids which can be produced in our body are called non-essential amino acids. We do not supply through our diet, whereas the amino acids which must be supplied to our food is known as essential amino acids. Essential amino acids cannot be built in our body. It should be supplied to our diet. Whereas non-essential amino acids can be produced in our body. Okay. So, any imbalanced diet can cause what? Protein disorders. Okay. You might have studied protein deficiency disorders in your previous classes. Okay. What makes the protein deficiency is the deficiency of amino acids. Okay, we should supply amino acids through our diet. Some essential amino acids through our diet. Okay, now let's move to the protein structure. Structure of proteins. Protein structure. So you have studied protein is made up of number of amino acids one after the other. They are joined by peptide bonds. Okay. So this is one amino acid, next amino acids, okay, third amino acid, likewise number of amino acids join to form protein. And this side, the first amino acid, this end will be what? Amino group. Okay. This side of the first amino acid, what will be facing out is amino group, whereas this side will be carboxyl group. Okay. So this is the N-terminal end of the protein. In the structure, we say what is N-terminal end of the protein. Here the first amino acid is, so the amino group is facing this side. This is the N-terminal end, and this is the C-terminal end of the protein. So I think it's clear what is C-terminal and N-terminal of protein structure. Then when we discuss about the protein structure, there are 4 levels of protein structure. The first level structure is known as primary structure. The first level of structure, protein structure, is known as primary structure. Primary structure consider protein as a rod-like structure. According to primary structure, protein is a rod-like structure. Its one end is N-terminal, other end is C-terminal. One after the other, amino acids join to form this kind of a rod-like structure, a line. Okay. Whereas the secondary structure, primary structure, secondary structure. Okay. You can imagine why there is such necessity. Okay. In a primary structure, protein is considered as a rod-like structure, rigid rod-like structure. In our protoplasm, in our ribosomes, a flexible, particular configuration is there. Okay. In our nucleus, there is protein. Everywhere the protein cannot remain as a rod-like structure like this. Okay. It was not at all flexible. What will do? It will remain as a very rod-like structure inside our protoplasm and wherever it is. So it is impossible. So there is a secondary structure of protein. It considers protein as a flexible structure where this is allowing some sort of folding. Okay, in secondary structure there is two structures proposed. One is alpha helix, other one is beta sheet, beta pleated sheet. Two structures for protein. The secondary level structures of protein, there are two types of structures. One is the alpha helix, which consider protein as a right-handed helix, N-terminal and C-terminal end. Because of some folding, it is forming right-handed helical structure, means then it is alpha helix. Okay, if protein, instead of remaining as a rigid line or rod, if is permitting some right-handed helical calling and if is existing like this is alpha helical structure. Many proteins exist in that form. Then beta sheet is allowing this type of a configuration to, parallel sheets will be there, this type of a configuration to protein, N-terminal, C-terminal. Okay, so in secondary level structure, the protein is with help of some kind of foldings, protein is forming alpha helical structure and beta pleated sheet like structure. Okay, instead of remaining as a rod like structure. Then the next level, tertiary structure of protein. Tertiary structure of protein. You just imagine again further calling happens here. Okay, it's again allowing more foldings. Then in tertiary structure, protein is considered as a woolen ball like structure due to high degree of calling. Okay, so high degree of foldings is allowed so that it is forming a woolen ball like structure. Then it is a tertiary structure. Okay, and one more level, this is not normally there for proteins, primary structure, secondary structure and tertiary structure. And the last level is applicable to only certain proteins. The quaternary structure is shown by only few proteins which is made up of more than one polypeptides. Okay. Normally protein is made up of one protein is one polypeptide. From here to there amino acids are joined. If protein is made up of like this one more or two more, okay, or again three more, Like this, so many polypeptides are there in the structure of a protein, means complex protein. A protein, if it consists of more than one polypeptide in its structure, it forms the quaternary level structure. Here, suppose this is one polypeptide, okay, then finished, next polypeptide, okay, then next polypeptide, okay. Likewise, here on a string, many balls are attached, many spheres on a string like. That means the N-terminal end of first one, it's C-terminal, next one N-terminal, it's C-terminal, next one N-terminal, it's C-terminal, next one N-terminal, it's C-terminal. That means this polypeptide ends here, next polypeptide start and ends here, next polypeptide start and ends here, this one ends, start and ends. 4 polypeptides are shown here. That means if in the structure of a protein, if there are many polypeptides, it gives an appearance of a string with many spheres. I mean a string with many woolen balls like this. Okay, if there are 4 polypeptides, 4 woolen balls like. Okay, then that is a quaternary level structure of a protein. This is what is about the structure of protein. It can remain in primary structure, secondary structure, tertiary structure and quaternary structure. Okay, then quaternary structure is applicable to protein with many polypeptides. An example is hemoglobin. Hemoglobin is formed of 4 polypeptides. Likewise, this 4 polypeptides are there. 2 alpha polypeptide chain and 2 beta polypeptide chains are there in hemoglobin. Hemoglobin is a protein made up of 4 polypeptides. 4 polypeptide chain, 1, 2, 3, 4. This can remain in this quaternary structure. It has 2 alpha chains and 2 beta chains of peptide, polypeptide. Okay, is it clear? If protein structure and details are clear, means we will stop for today and meet you in the next class. Thank you.